here in the Labdia Labor Diagnostics, we have many departments and they work on the different topics and there are experts there, and we wanted to have a system that bring them all together in one lab information system so that we can do sample tracking but also write reports together, see other results of other departments and so on.
At the St. Anna's Children's Cancer Research Institute, research is conducted simultaneously in different laboratories in fields of hematology, infectiology, immunology, oncology, and human genetics. Due to the use of different methods such as NGS, Sanger, FISH, MLPA, FACS, and many, many more, the demands on the LIMS are very, very high. Complex sample distribution, many different methods, high sample throughput, and different workflows are just a few examples. We are currently in the middle of the implementation of the project and asked Dr. Marie Bernkopf why St. Anna CCRI chose the donor genetic LIMS and what was particularly important to them in their choice.
We are here at the St. Anna Children's Cancer Research Institute in the heart of Vienna. And here we are working on cancer in children and adolescents. And a subsidiary of the St. Anna Children's Cancer Research Institute is the Labdia Labor Diagnostic. And here we provide diagnostic services in the field of hematology, oncology, infectology, human genetics, and immunology. The lab information system from Dorna that I made gave us this flexibility to have these different departments and different requirements for every department and bring everything together in one system. And this is exactly the flexibility we needed.
Were there any features that were particularly crucial for your laboratory?
So for us, that was definitely the work lists and the batches. So we work a lot in batches as well in our lab. So several samples are processed in the same way in the lab. So to have the feature of the batch is great for us. So we could upload documents there, write down if the QC was okay, and then send all the samples that were processed at the same time, when the QC was okay, then into the analysis, for example. So this is now all digitalized in our lab and with the IMID.
How long did it take for your team to become fully familiar with the system?
We are still actually in a kind of adventurous phase where we find certain features that we also discuss with our admins of the system if we can implement that for our specific workflows. So I think we are fully trained in the daily work we are doing in the system, and that took a few weeks, I would say. So it wasn't that long because the things you do on a daily basis, you do so often, you will learn it quite quickly. But for new features, I think we're still in a learning phase there. And it's good that the IMID has so many new features that we can use.
And last question, would you recommend donor genetic LIMS to other laboratories?
I would definitely recommend the genetic LIMS from Dorna to other users, simply because it's a very flexible system where you can mix and match features and masks, and even buttons have exactly how you want them. And this flexible system is perfect for this very complex area of genetics.